Next up, we have chia seed. Sorry, chia seed.
Yeah, hello everyone. Today I'm going to introduce my PhD project, the transmission of bacteria-resistant genes in aerosol. Antibiotic-resistant genes are spreading worldwide, and it can be detected in the water, soil, and even in the air, and they can be transported through the aerosol. But here the thing is that the resistant genes are already existed in the environment. But what's really matter is if the bacteria can pick up these genes, which means, in other words, if it's like the aerosol, it just work like the trunk to transport the resistant genes from one place to another place, or it also provides the active environment to help the bacteria to pick up these genes. And if it really happened, it implicates the aerosol will play a more important role in the gene transfer. Okay, let's give a brief introduction about what is a gene transfer. Okay, so here we have a bacteria with resistant genes, and we call it donor. And also we have a bacteria without its gene, it's empty, so we call it recipient. And when they come into contract, the gene transfer happen. The resistant genes will transport from one bacteria to the recipient, and now we call this recipient acquired the resistant genes. We call it the transconjugates. And here we also have a number to quantify the possibility how the event will be happen. And here we call the conjugation, like the gene transfer efficiency in here is a number of the transconjugates bacteria divided by the number of donor we're providing here. Okay, so keep going. Basically, in the bulk phase, we already know that gene transfer can be happened in the bulk liquid, like the gene transfer could happen between the bacteria, and also we already know like there's a lot of factor like the oxidative stress or the temperature or the relative humidity change will also affect the gene transfer efficiency. And also, like the oxygen will affect the abundance of the gene antibiotic resistant genes in bulk phase. And don't you think these factors is just a similar like what we have in aerosol phase? And moreover, even in the aerosol phase, we can have a more extra environmental stresses like the UV exposure or the pH change or the carbon dioxide concentration will also give a lot of extra stress to help the maybe. So here we think in the aerosol phase, it will have a potential to help the bacteria pick up the genes. We call it promote the gene transfer happen. But here's also have some challenges in here. As we previous have we investigated so far in the aerosol environment, there's a lot of the physical and chemical stresses like dehydration or the oxidative stress. The bacteria survive. It will die very quickly in the aerosol phase. It looks like it will only like 99% bacteria will die after 1 hour. There is a very tough question. And also, because of the gene transfer itself, it's a very rare event. Because it have a very low efficiency, and it's need even more 2 hours to make the things happen. Like the the conjugation efficiency is only like 10 to minus 6 or 10 to minus 5. It's quite low. So this is a problem we want to fix it, and to achieve our aim is to see if the gene transfer can occur in the aerosol environment. Okay, so move to our first part. We need to optimize the gene transfer conjugation efficiency in bulk to help us to observe the possibility the gene transfer happen. So here is how the figures looks like. In bulk phase, so here we picked up the where objective is like the donor is an E. coli with a tetracycline resistance, and also the recipient is a nalidixic acid resistant. So which means if the gene transfer really happened, the new transconjugated bacteria will have both two antibiotic resistance. So in this case, we can use a selective plate to pick up the colonies on the plate. And so here we tried a lot of different different factors to see if it will help the conjugation happen in the bulk phase. So you can see here, we tried a lot of different factors, but in here, the optimal condition, the best environment to support the things happen is here. We concentrate the bacteria over the 20 times. We concentrate and do a centrifuge to pick the pipette. And also we tried the higher salt concentration, like 2.5 times salt, the concentrated solvent in here. And also a lot of different conditions. So in one sentence in here, we have promote the gene transfer efficiency from 10 to minus 6 to 10 to minus 4, which means we enhance the efficiency over 100 times in here. That is a good thing. And based on the equation we showed at the beginning, what does that mean? Which means in the past, at least we need 200,000 bacteria to support us to observe this phenomenon happen. But now we only need 2,000 bacteria. This is a very good thing to help us to see it. Okay, but there is another question happen. We know here we need a live bacteria is about 200 bacteria in here. So based on that, we can do a simple calculation, which means we need like 400 droplets to support the following things gene transfer to happen. And here is the slabs. I think you guys have see a lot about that. We have introduced all that. This is a very good equipment to help us to have a control of the single droplets in the precise environmental condition. But here is a limit that the maximum droplet they can hold is just 15 droplets. It's not match our specific requirements. So keep going. What we do is, we update our slabs to QLabs. This is how it looks like. Basically, the simple idea is that we change the inner structure to the 2 electron rings to the 4 quarks in here, which means we have more space to hold the droplets and leave it for the longer time. And here is a video how it works in here. Have a look. So here we generate the droplets and transfer in the middle of the electron, and then you can extract part of them at any time point that you want. This is how it works. And and keep going. We have do a lot of the feasibility tests to test how exactly the droplets we can have in here. So this picture shows we can have a precise control of the number of droplets from several droplets, like the 8 droplets from over 200, and even now we can have the max over now we can have over 500 droplets in here. This is a good thing, which means it can meet our specific requirements, and also we have the feasibility test to see. You can hear we treat with 2 different concentration of bacteria solution, and here shows a very linear relationship, which means the bacteria can distribute evenly in each droplet. So in here, we can see the QLabs provide us over 500 droplets in here, which means we can, if we have 30 bacteria per droplet, we can do a simple calculation, which means we can have over 50,000 bacteria in here. And also we can have the levitation time for even 4 hours or even more. And now basically we can do the levitation for overnight. This is a very strong equipment, and there is a lot of future beyond this. And if you are interested in, you can come and talk to me about later. Yeah. Okay, cool. we have solved the system problem. This is a good thing, and keep going. In the aerosol phase, we want the bacteria live longer. Okay, so here, basically you can see this black column is in the normal condition. Almost, after 1 hour, almost 99% bacteria will die. But if we treat them, you can see this orange column in here. If they treat with a higher salt concentration, like 10 times the concentrated solvent in here, it can increase the viability from only like less than 1% to 40%. And this is a very good thing to help us to help the bacteria live longer. And also we tried with the bacterial life cycle. We do a lot of the testament, try a lot of different conditions. But okay, in here we can see we treat them with a high salt, give another stress in here. The solvent concentration is 5 times LB will promote the better protective of the bacteria, and also tried the relative humidity, different relative humidity from 30% to 90%. We found, okay, the optimal condition is 85%. And also we tried the antioxidant against the oxidative stress, like the glutathione is a better choice. And also we tried a different bacterial life cycle, and we have the stationary phase is our better choice. Okay, so in total, give a rough idea is that this is how the bacterial viability curve looks like before. But after we spend a lot of time to optimize the condition here. At the end, we can see we can increase the bacterial viability from 10% to 65% after after 1 hour. So this is what we achieved so far. So okay, we bring the two parts together. So which means we can enhance the bacterial viability, and also we increase the gene transfer efficiency. But the whole idea is that to help us to observe if the gene transfer could happen, we want to enhance the possibility. But here, this is just the modeling things, but there is still a lot of unknown things we need to figure out, like the time mismatch limits the gene transfer detection. Okay, in summary, what we spend time to optimize the gene transfer in bulk phase, and also we optimize how long it lives in the air. So this we take these things together, and the further step is we want to see what will really happen in the aerosol, and that is our aim of this project. Okay, so at the end, many thanks to my supervisor Alan and Jonathan, and many thanks to Bristol Aerosol Research Group, and also about Bio Aerosol Group. Yeah, thank you. That's all.
Thank you very much. Yeah, we have time for some questions.
David from Grommet. So to kind of summarize it, I'm not a biologist, but is there an optimization or balance between if you keep the little guys in the air happier, they're less likely at the end to have to develop microbial resistance genes.
Exactly.
But if you kill them all, they can't do it. So somewhere in the middle where it's an optimal point for them to develop this. And is that then the intention to kind of have the knowledge of, then you have to relate that to how natural aerosols are emitted in, say, a hospital setting, and try and avoid that at all costs, because that's the
that is the whole idea.
But the brain's problem.
Yeah, got it. Yeah, that's very good. Yeah.
Great thought, thank you very much.
Yeah, thank you.
Adam, well, very nice talk, thank you. So you've got a higher what's it called, infection efficiency and droplets. You say, could this be a concentration thing, like when water's evaporated, then there's just like more bacteria closer together?
Yeah.
Is the stress you separate two effects?
Yeah. This is a very good point. Basically, we have tried like the concentrated bacteria and do the levitation in the aerosol, and we did see for if the bacteria more concentrated, it will help their survive in the aerosol. So which means okay, that maybe the conjugation or gravitation or the water evaporates is really help their survive, provide a protect shell to help them to alive better in the aerosol. Yeah.
And also the infection, is that more or less concentrated?
Yeah. That is. Okay, thank you.
Maybe time for one more question. Okay, so let's move to the next speaker. Thank you very much, Jan.